in length, the Re increases. Consider figure 6.13, which shows patterns of flow downstream of a cylinder under conditions of varying Re. When Re is about 30, a pair of attached vortices reside just downstream of the cylinder. This might give a small coral the opportunity of feeding on particles that are relatively stationary. As the coral grows larger, these vortices become more erratic, however, and food may not be held in a predictable pattern. Size and velocity can increase together. As an organism grows larger, it often projects into a rapid mainstream current. The use of flumes. Flumes are useful for studying the effects of moving fluids on organisms, although flumes must be scaled carefully. It is usually quite tricky to study the effects of flow in the marine environment. Some clever investigators have devised field current meters and have been able to characterize the flow field around an organism. Because of inaccessibility, this becomes impractical for studying the streamlining of a tuna or the flow about a deep-sea organism. Even in accessible habitats, it is very difficult to measure flow on the smallest of scales. Electronic devices, such as flow meters based upon thermistor sensors, are usually difficult to use accurately in the field. In still waters, microcinematography allows us to study the behavior of very small creatures at low Re. In moving waters, flumes of various types are used to study the effects of flow. A flume is a device that includes a source of moving water, a working area where the organism and flow field are characterized, and a drain-return system. Most flume designers seek two objectives: maintenance of laminar flow and maintenance of scaling by Re. A long flume is desirable because it takes a while for the flow over the bottom surface to stabilize and produce a predictable boundary layer and velocity profile above the bottom. A wide flume, relative to water column height, prevents effects of the walls on flow. Scaling by Re is also essential to keep the proper ratio of inertial to viscous forces. This refinement has an advantage, however, in that you can study a very small object, such as a copepod, by making a larger model and placing it in a more viscous medium. Using a flume, one can study the hydrodynamic forces at work on a biological object. For example, I have studied the reaction of the siphon of a sediment-ingesting bivalve mollusk, Macoma secta, which feeds much like a vacuum cleaner. At low mainstream current velocity, the siphon is protruded into the water and swirls around, picking up sand grains. If the current increases to about 15 centimeters s, the pressure drag on the siphon makes the siphon difficult to control, and it is withdrawn. The animal then feeds on sediment within the burrow. At velocities above 35 centimeters s, the bottom sediment is stirred up and the bottom is very unstable. At that point, the animal ceases to feed. Using a flume with a video recording device allows such qualitative observations quite easily. Chapter 7: Reproduction, Dispersal, and Migration. Reproduction, dispersal, and migration are the fundamental processes that allow living populations to grow and exploit new habitats. Reproduction is the replication of individuals and is necessary for population growth. Nearly all species have some form of sex, which allows exchange of genetic materials among individuals. Sex and reproduction are intimately related, but many organisms can reproduce without sex, so the two processes can be discussed separately. Dispersal is the spread of progeny from one location to another, usually to locations that differ from that of the parent. Dispersal is a one-way process and often is controlled by the vagaries of water currents. Because water is a supportive medium and because water circulates rapidly over great distances, many newly hatched young can disperse over large distances, often as much as hundreds of kilometers. As a result, marine species often have broad geographic distributions and the capability of rapid extension of their geographic ranges. Migration, a directed movement between specific areas, allows an expansion of types of habitat use and can increase a species' efficiency at exploiting the best resources at optimal feeding and spawning sites. Ecological and evolutionary factors in sex. Costs and benefits of sexual reproduction. Sex is a nearly universal characteristic, despite the considerable costs to organisms of maintaining it. Sex is a species property whereby different individuals have the capacity to exchange or combine DNA, which causes offspring to differ genetically from their parents. Sex may involve simple transfer of DNA between bacterial cells or the union of gametes to produce zygotes. Nearly all organisms have some form of sexuality, ranging from the mating types of bacteria to the separate sexes and mating dance of humpback whales. The near universality of sex is a paradox because of its considerable cost. In a typical diploid organism, each parent contributes the same number, n, of chromosomes to the offspring, giving the proper diploid number of 2n. However, the mother usually invests much more in her offspring than does the father, even though half the offspring's genes are his. Why not devote all her energy to nurturing offspring that carry only her own genes? Otherwise, she invests a lot to place only half of her genes in any offspring. There are other costs to maintaining sex. Finding a mate can be costly in terms of time and energy. In fiddler crabs, the males have 2 claws, one much larger than the other, which comprises over 30% of the body weight. This larger claw is used only for sexual displays and for combat with other males. Because the claw is not used for feeding, males must compensate for this handicap by feeding longer and faster. In one form of sexual display, the male's major claw is stretched out to the side and the crab has exaggerated musculature and stouter legs to support the weight. Why should so much cost be attached to sex and sexual differences such as these? The most obvious benefit of sex is the generation of genetic diversity. Genetic variation allows offspring to survive in a broader variety of habitats. There is also an increased potential for evolutionary change when the environment changes. Without sex, all offspring are genetically identical to the parent. No evolutionary change is possible except by means of mutations, which occur only rarely. Asexual populations are, therefore, clones, with little or no genetic diversity, except by mutation within the clone. In contrast, sex provides continually new combinations of genes and changes of gene arrangements due to chromosome crossover during meiosis. The process of natural selection results in the eventual dominance of the adaptively superior genetic variants. A study of the snail Potamopyrgus antipoderum demonstrates how genetic variation allows adaptation to the challenge of disease. This species, living in New Zealand freshwater, has populations that reproduce sexually and asexually. The asexual populations are far less able to resist invasions of parasites. Sexual populations grow more slowly than clonal populations, which accords with our assumption that sex does have a cost in population growth. The continuous evolutionary battle between snails and their parasites results in many local sexual populations that are each adapted to different suites of parasites. Sexual selection increases differences between the sexes and is driven by mate selection and polygynous behavior patterns. In most animals, males compete for mates. Females invest more energy in gametes and energy in rearing offspring. Males invest in form and tactics that maximize the number of matings they can achieve. Once sex exists with inter-male competition, the differences between the sexes can be enhanced by sexual selection, the selection for male secondary sex features that increase mating success. Genetic variants that have the most successful mating abilities will increase in the population. For example, fiddler crab males with larger claws may be better at attracting mates or holding high-quality territories where females can incubate eggs. This superiority would set in motion a selective process that could lead to selection for larger claw size. Success in sexually selected characters can result in occupation of high-quality habitats by means of inter-male combat, where matings are gained and winners emerge in a process of intrasexual selection. Alternatively, conspicuous coloration in males might reflect their high fitness, which may be used as a criterion in mate selection by females, also known as intersexual selection. While such selection may result in more extreme structures, trade-offs with function may prevent a structure from becoming too extreme. In the case of fiddler crabs, too large a male major claw might increase fatigue as males run from predators. In many species, selection for bright male color might be countered by increased detectability by visual predators. See Hot Topics Box 7.1 for more on this subject. Intersex differences are also driven by social structure in mating patterns. In many species of fish and mammals, one male may mate with a group of females. A male elephant seal is about 5 times the mass of a female and may inseminate a group of 10-100 females during the breeding season on the beach. The male's dominance over other males first involves vocalizations toward other males to establish dominance, but often escalates to bloody battles. If the ability to be dominant is correlated with fitness, then the female gains an advantage in the fitness of offspring she produces. Types of sexuality and their value sexes may be separate, simultaneous, or sequential in the same body. Organisms of nearly all kinds have 2 sexes. Species that have separate sexes, or gonochoristic species, are the most familiar. While males may mate in direct contact with females, seawater permits many species to simply shed sperm into the water. Planktonic gamete transport often involves simultaneous spawning of males and females, which is often keyed into tidal or lunar rhythms. A hermaphrodite can produce gametes of both sexes during the individual's lifetime. In simultaneous hermaphrodites, sex cells for both eggs and sperm are active within a single individual at the same time. Despite this, self-fertilization is rare, probably because inbreeding often produces inviable offspring, which imposes selection against such behavior. Being a simultaneous hermaphrodite means never having to say you're sorry, because an encounter between any 2 individuals guarantees that mating can occur. For example, acorn barnacles need to copulate to reproduce, but they are stuck with a sessile lifestyle and can reach only nearby barnacles. Simultaneous hermaphroditism is a great advantage because the nearest individual will always be of the complementary sex. An extraordinarily long penis allows a barnacle to reach relatively distant fixed mates. Acorn barnacles often have a seasonal cycle of growth and degeneration of the penis. They show phenotypic plasticity and can grow a longer penis if a potential mate is far away, or can even grow a more strengthened penis if the local microenvironment is more turbulent. One of the most fascinating aspects of simultaneous hermaphroditism is the common continual trading of mating roles. In many simultaneously hermaphroditic fishes, for example, members of a mating pair change sex roles every few minutes, first producing sperm, then spawning eggs. Sequential hermaphrodites start their mature sex life as one sex, and then transform into the opposite sex. If they are male first, they are said to be protandrous, whereas if they are female first, they are protogynous. Many invertebrates, including some oysters, polychaetes, prawns, and coelenterate, are protandrous, whereas some fishes, particularly coral reef fishes, are protogynous. In some cases, such as the diminutive protandrous polychaete species of the genus Ophryotrocha, sex change can involve a mere switch from the manufacture of oocytes to the manufacture of spermatocytes. In protogynous reef fishes, however, the sex change also involves color, size, and morphological transformation. The snail Crepidula fornicata usually occurs in stacks, with larger and older females below and smaller and younger males on top. Top layer members of the stacks are oriented with right anterior margin in contact with the same margin of the lower member. This allows insertion of the penis of the upper snail into the female gonopore of a lower snail. The stimulation to change sex involves contact with other individuals. To change to a female, a male must be in contact with a female below in the stack. The relative contributions of different sexes to zygotes in the next generation determine the value of hermaphroditism and the relative sizes of males and females. Why be a simultaneous hermaphrodite? In a sense, one might turn the question around and ask, why aren't all organisms simultaneously hermaphroditic, given the obvious advantage of finding a potential mate in every individual? The answer may lie in the limitations of being a jack of all trades and master of none versus being a specialist. Simultaneous hermaphrodites must invest not only in an apparatus to form gametes but also in the secondary sexual attributes necessary to attract another mate for both sexes. If one started with a population in which genetic variants for hermaphroditism and for separate sexes coexisted, one might find that sexual selection would favor genetic variants for pure males and females. The hermaphrodite would lose out in the competition because it serves both functions but each less efficiently. Hermaphroditism is favored only when the value of always being able to find a mate compensates for the disadvantage of not being a sexual specialist. The acorn barnacle may be just such a case. Simultaneous hermaphroditism is far more common in freshwater invertebrates, where it is generally more difficult to find a mate owing to the lower population densities and the uncertainties of freshwater existence. Sequential hermaphrodites would seem to have all the advantages, given that they can be both sexes at different times. Unlike simultaneous hermaphrodites, however, they lack the advantage of always being able to find a mate in another individual. Then what is the advantage of changing sex? The answer lies in the advantage of being a male or female at different ages or sizes. Consider protandry, or the quality of first being a male and later a female. As a general rule, producing eggs costs more energy than producing sperm. It is also usually true that a larger and older animal has more energy at its disposal. Then it would be best to be a male while still small, when a relatively small investment in sperm could yield many offspring. Above a certain threshold size, however, females can parent more offspring because the available energy can be used to produce more eggs. Male success would reach a plateau with increasing size. This is known as the size advantage model. The threshold size of switching should be that size at which the number of offspring parented would be the same regardless of the sex of the animal. Below the threshold size, the animal would sire more offspring if it were male. Above that threshold size, it would parent more offspring were it a female. The considerations are thus simply those of a cost-benefit game. This model was tested in a protandrous species of the polychaete Ophryotrocha. Larger males were less successful in gaining mates than smaller males, whereas female reproductive output in females continued to increase with increasing body size. These results favor the hypothesis that size is the advantage that selected for the switch from male to female function. Why then should an organism be female first, then male? Reproductive success as a male often involves intermale competition, involving combat, bright colors, and rapid swimming. Most of these traits are enhanced with increased size and experience. Many territorial coral reef fish have flashy males and are protogynous. For example, the cleaner wrasse labroides dimidiatus is usually found in a group of 10-15 fish. All but the largest fish in the group are relatively dull in color and are females. By contrast, the largest fish is brightly colored and male. The male constantly tries to prevent the females from mating with interlopers. If this male is removed from his harem, the largest female will transform into a male. In other species of wrasse, individuals can either be terminal phase males, which hold territories, initial phase males, or females. Initial phase males do not hold territories and either spawn in groups with females or attempt to sneak into the territories of the terminal phase males and gain matings. Both initial phase males and females are capable of changing into terminal phase males. The proportion of initial phase males varies from place to place. This suggests that there are places where being a small male results in more successful matings. Although it is possible to classify types of sequential hermaphroditism, it is important to realize that many natural populations contain considerable geographic variation in sex change. This is especially true of coral reef fish. Certain species may be locally protogynous but gonochoristic in other sites. Hermaphroditism is not necessarily a fixed trait of a species and may be under active natural selection in different directions in different populations. Male morphology has been found to be dimorphic in some species. Kurdziel and Knowles examined male dimorphism in an amphipod. Some males had a large structure known as an athapod, which was used in aggression, but other males lacked this large structure and more resembled females. While the aggressive males guarded females until they molted and were ready to mate, the other males were sneakers and approached unguarded females. As it turns out, the sneakers were as successful as the large aggressive morph in getting mates, which explains why the male dimorphism is maintained over time. Male dimorphism is found in many other species, including several species of rockpool blennies, which have a nesting male morph, who attracts females to lay eggs in the male small territory, which are cared for by the nesting male. But an alternative smaller male morph can sneak in and achieve matings within the nesting male's territory. Dwarf parasitic males are found in some species that live in situations where it is difficult to find mates. Where the sexes are separate and mate location is very difficult, males may be small animals that either attach to larger females or reside very close to them. Good examples are found among some barnacles, where tiny dwarf males attach within the mantle cavity of normal-sized females, e. G. Species of the stalked barnacle Scalpolum amibia and the boring barnacle group Acrothoracica. Many deepwater fish have dwarf males. Males show varying degrees of modification and may be parasitic upon females. In some cases, the male is a miniature version of the female, except for the reproductive organs. This is common in some brittle stars, whose dwarf males cling to the much larger females. In some cases, dwarf males of deep-sea anglerfishes find and attach to females and even have their blood circulation systems in connection with each other. A rare case of eusociality can be found in groups of alpheid shrimp in spaces within large sponges, where one female is served by a large number of males. When individuals are closely related and found within an isolated space, one might expect a division of labor that serves a genetically closely related group. In ants and bees, all members of a colony are closely related, and such division of labor into queen and different types of workers can be found. Why is there such altruistic behavior, in which workers serve the queen? William Hamilton noted that workers are extremely close relatives of the queen. Their role in doing the work of the colony, therefore, increases not only the genes of the queen but also their own. The evolutionary force leading to the evolution of cooperative behavior among close relatives to proliferate similar genes is known as kin selection. The extreme case of an entire colony of individuals behaving in cooperative fashion is known as eusociality. This behavior usually is found in a group of individuals that serve a queen's reproduction. It is extremely rare beyond social ants, bees, wasps, and a few other groups of insects. Naked mole rats are also eusocial. There is one queen, plus several mating males and numerous other closely related non-mating siblings or very close relatives that gather resources and take care of the tunnels in which the mole rats live. A very similar case has been found in some species of synalphyte shrimp, where large sponges in the Caribbean have colonies each with one female and 100-200 males and juveniles. When snapping shrimp from outside the colony are placed near the opening, males swarm to defend the colony, attacking the intruders and killing them. The shrimp recognize members of their own colony and can even signal to each other, warning of intruders. J. Emma Duffy, who investigated these shrimp, found through genetic analysis that individuals of the colony were closely related and the relatedness was consistent with the individuals being siblings. Thus the snapping shrimp fit the condition of closely related individuals in a confined space, all serving the colony by a specialization of function, with the single female being an analog of the queen bee. Fertilization success is affected by the mode of sperm transfer, the volume of gamete production, the distance between males and females, water turbulence, timing, and behavior. Many animal species use a variety of specialized means of sperm transfer involving direct male-to-female contact. In the simplest cases, found in some polychaetes and fishes, the male applies sperm to the body surface of the females or to an egg clutch as the eggs are released by the female. Males of many species have copulatory structures. Many species, ranging from migratory fishes to intertidal drilling gastropods, form breeding swarms at the time of mating to further ensure the finding of a mate. These modes of sexual contact increases the probability of fertilization and allow for mate selection. Although direct contact and copulation guarantee sperm transfer, many marine species take advantage of the presence of water and shed their gametes directly into it, which is known as free spawning. At best, the gametes will mix and fertilization frequency will be high because the sperm are so much more numerous than the eggs. However, turbulence or large distance between spawning individuals greatly reduces fertilization success. We discussed the costs of free spawning below. Spawning by one or a few individuals may induce mass spawning of an entire local population. Mussels will spawn in response to phytoplankton in the water. The mussel larvae then can eat the phytoplankton. Timed spawning is usually keyed into lunar tidal cycles. In some reef sponges, all male individuals spawn simultaneously, spreading a fog of sperm over the reef. Many polychaete annelids can change morphology radically to transform themselves into epitokes, individuals that are essentially swimming sacs of gametes. Operating on a lunar cycle, they may swim to the surface and perform a nuptial dance, in which males and females swim rapidly about each other while releasing gametes into the water. South Pacific peoples take advantage of this behavior by harvesting swimming polychaete pololo worms. Many coral reef fish shed their gametes, but the fishes often locate themselves strategically on the downstream end of patch reefs to minimize turbulence and gamete dilution. As a result, fertilization success is surprisingly high, often exceeding 90%. Free spawning has a number of costs relating to turbulence, polyspermy, timing of spawning, and avoidance of interspecific hybridization. With free spawning, the game changes because both eggs and sperm must encounter each other, sometimes based upon hygrodynamics as opposed to sex appeal. In free spawners, the eggs suddenly are part of the gamete collision equation, and there might be strong selection for egg traits that maximize sperm encounters and even attraction. But there are also significant costs of free spawning. 1. Fertilization success. Fertilization success is often low when distance between males and females is great and when turbulence is strong. At low population densities and with few spawning individuals, one might expect to see the effect of sperm limitation, and this has been demonstrated in the eastern Pacific temperate sea urchin Strongylocentrotus franciscanus. Some species, such as the Pacific sea star Acanthoster plancy, produce such large volumes of gametes that high rates of fertilization can occur between males and females separated by tens of meters too. Timing of egg and sperm release. Gamete release of both sexes must be simultaneous for fertilization to be successful. Gametes often do not last much more than a few hours, meaning that an environmental signal to ensure simultaneous spawning, such as a response to a lunar cycle, must be evolved. We discuss mass spawning in coral reefs in Chapter 17, where many species queue into lunar cycles. Alternatively, some species of mussel are known to respond to the spawning of nearby individuals. This may mean that certain local individuals will make a disproportionate contribution to the next generation, especially when a male invertebrate spawning millions of sperm successfully fertilizes a large number of gametes of nearby females. Because water turbulence should dilute gametes and reduce the percentage fertilization, we might expect that some species would tend to release gametes only in quiet water, when the turbulent dilution effect would be minimal. The seaweed Fucus vesiculosus releases gametes only at current speeds of less than 0.2 ms. At these low speeds, usually occurring in the early evening, fertilization success is nearly 100% in the lab. Experimental increases of water speed causes the seaweed to reduce its release of gametes. 3. Avoidance of interspecific fertilization. What if several species evolve a response to the same lunar cycle? Many species would be spawning at the same time. Gametes would likely encounter those of other species. If reproductive isolation has occurred, this would cause lowered fitness unless there was extreme specificity of gametes for fertilization at the microscale. In quiet water, many sperm may simultaneously encounter a single egg. High population density of males might select for more sperm production because male genotypes are competing with each other for matings, and increased sperm release might increase the production of successful fertilizations. So more sperm is better. Or is it? If sperm concentration is very high, then polyspermy, which entails the beginning of zygote formation by 2 or more sperm and 1 egg, may develop. When this happens, successful fertilization is usually foiled. All known animals have elegant blocks to polyspermy, some of which act within seconds to prevent another sperm from attaching to the egg. But the phenomenon is still possible and perhaps likely in a concentrated cloud of sperm, as might be produced by some invertebrates. 5. Fertilization and small-scale turbulence. On the scale of meters, turbulence and current flow are essential for gamete encounters. Otherwise, gametes would sit still. But, on the scale of millimeters and less, turbulence also is very important for fertilization. With no turbulence, gamete encounter would be reduced because of the problem of viscosity generated by low Reynolds number. So a small degree of turbulence increases gamete encounter and fertilization success. But when turbulence increases further, gametes will not have the time and opportunity for sperm-egg recognition to occur and fertilization rate will be reduced. There are 2 main phases in small-scale gamete encounter: attraction on small scales of sperm to eggs by means of sperm attractants, followed by surface compatibility of sperm when attempting to fertilize eggs. Sperm are usually motile, using the flagellum to swim. But on scale of millimeters, sperm in the neighborhood must detect the presence of eggs, and, of course, eggs might signal their presence. To accomplish this, many species use sperm attractants. For example, eggs of the Pacific coast red abalone Haliotis rufescens release the amino acid 1-tryptophan. When abalone sperm encounter a threshold concentration, they swim toward the egg, which is now just a couple of millimeters away. The egg is surrounded by a chemical plume of this sperm attractant, and the sperm probably encounters this plume randomly. The chemical plume increases the target size that the egg can present to recruit a sperm cell. Sperm attraction is also known for seaweeds. In the laminarials, pheromones are produced by a microscopic female gametophyte stage, which induces male gametophytes to release sperm. The pheromone lamoxarine may exist in several isomers, but there does not appear to be specialized species recognition based on difference of gamete recognition between species, as we shall now discuss for some animal sperm-egg recognition systems. In some groups, specialized gamete recognition proteins are employed to avoid interspecies fertilization. Because different species will be selected to spawn when gamete contact is optimal, at a specific time when simultaneous. Spawning of males and females can occur in turbulence is low enough that gametes can collide. It is to be expected that different species might have gametes come into contact. So, is there a mechanism that keeps gametes of different species from attempting fertilization? We can see several steps at fertilization that might be disrupted when gametes of different species come into contact. 1. Receptors in the egg coating induce the sperm acrosome reaction, which is an initiation of attachment. 2. Sperm and egg attach, owing to the presence of complementary binding and receiving proteins. 3. Further reactions cause the uniting and fusing of gamete plasma membranes. We have information on only two major groups of organisms, mollusks, especially abalones, and sea urchins. But a surprising similarity has emerged. Both have a highly specific sperm-egg binding system. In urchins, sperm use the protein bindin, which unites with a bindin receptor in the egg membrane. In abalones and other mollusks, gamete recognition is controlled by the sperm protein lysin, which creates a hole in the egg envelope, but only for eggs of the same species as that of the sperm. In the abalone egg envelope, a receptor binds specifically to lysin. Hybrids are thus rare, even though 7 species of California abalone freespawn gametes and overlap in geographic range and breeding season. Different species of urchin rarely, if ever, have gametes that will fertilize each other. Bind-ins from different urchin species are different proteins and, therefore, have different amino acid sequences, and this makes the crucial difference in the sperm-egg recognition process. The same is true for lysins in abalones. Proteins are typically folded in a complex manner. Some of the amino acids are involved in regulating the three-dimensional structure, while others are involved in binding with other molecules. Thus, only a portion of the amino acid chain is involved in gamete recognition. One of the fascinating aspects of these gamete recognition proteins is the apparent high rate of evolution of amino acid sequences compared to other proteins. The accelerated rate might be due to a response to evolution in the recognition protein found on the complementary gamete. For example, the bindin protein found on an urchin sperm binds with a protein receptor known as VRL. There may be a constant tugging in evolution between the two proteins. When one evolves, then the other must evolve as well. Stephen Palumbi found that there is genetic variability of bindin sequences within populations of species of the urchin genus Economitra, and that eggs may be selective for sperm with some bindins relative to others. This is a form of sexual selection that may drive the evolution of bindin and VRL proteins. For example, when sperm are abundant, rare bindin genetic variants apparently are selected and increase in the population. When sperm are rare, it is the common genotype that is likely to have success in fertilization, simply by virtue of its high frequency. However, not all urchin species have this sort of polymorphism. Ultimately, if two populations are separated, the evolution of bind-ins can lead to complete incompatibility between species. Complete gamete incompatibility in sea urchins can be accomplished with as little as 10 differences in amino acids, and this amount of change can occur in as little as 1.5 million years. So, the mechanism of speciation in free-spawning species may be very much tied into the dynamics of protein evolution of these binding proteins in many unrelated species groups. Sympatric groups of closely related species of sea urchin, for example, have been found to have comparatively rapid rates of evolution of bindin when compared to species that are geographically separated from their closest relatives. This suggests that bindin evolutionary rate may be setting the pace for the evolution of reproductive isolation between species. Parental care is non-existent in many marine animal species, but in some cases females or males care for eggs, embryos, and young. Marine species often exhibit absolutely no parental care. Many of these species spawn eggs and sperm, which unite to form zygotes, which, in turn, develop into free-swimming larvae that are completely at the mercy of the seas. However, in some groups, such as the fiddler crab UCA, the female incubates the eggs for a couple of weeks and then releases swimming larvae into the water. Many species care for their young to an even greater degree. In some species, the young are reared to a juvenile stage within the mother. This is, of course, true for all marine mammals, but it is also true in invertebrate groups and fishes. The Atlantic and Pacific intertidal clam Gemma, which is only a few millimeters long, nurtures its young within the mantle cavity and releases them as shelled juveniles. The Chilean oyster Ostrea chilensis broods bivalve larvae within the mantle cavity and can tell the difference between its own young and other particles of about the same size, which they reject. In some species, courtship is strongly related to male parental care. Conceptually, male care should be linked to the assurance that the offspring are its own, and the investment in care therefore increases the probability of survival of the male's offspring. Seahorse mating pairs engage in a complex mating dance, often intertwined by their tails. Eventually they face each other, and the female releases eggs as the male opens up a pouch. The eggs are then fertilized and deposited within the male pouch, where they are then nurtured for 2-4 weeks, until they hatch and are released. The male of the three-spine stickleback Gasterosteus aculeatus has a vivid red belly during courtship. This anatomical feature serves to lure females to lay eggs in its nest, usually hidden among rocks on the bottom. Many aquarium enthusiasts have observed the male dance in a zigzag motion to lure the female to the nest. The great behaviorist Nico Tinbergen showed that the reproductive females have a stereotypical response. They are strongly attracted even to silver discs painted red on the bottom. Behaviorist Susan Foster has observed that in some locations, the female participates, perhaps to a dominant degree, in mate selection and often swims up rapidly to a male and jumps onto his back. During the courtship process, a female enters the nest and lays her eggs, which are fertilized externally by the male. Subsequently, the male aerates the eggs until they hatch. It is in the male's interest to rear the young of all females that have laid eggs in his nest because they are his progeny. When a female enters, however, the eggs that have already been laid there by other females are, well, just food. Commonly, an intruding female will gobble up the eggs already present and bolt. Non-sexual reproduction permits the same genetic type to increase rapidly in an open environment. Although sexuality is nearly universal in marine organisms, many of them are capable of reproducing without the formation of a zygote. Asexual reproduction lacks the cost of sexual reproduction and permits the spread of a genotype that has successfully colonized a given habitat. A population of genetically identical individuals, all deriving from one founder, is known as a clone. The exact style of asexual reproduction varies, depending upon the biology of the individual group. Diatoms, a major group of marine phytoplankton, consist of cells or cell chains and can reproduce asexually by fission. In multicellular organisms, fragmentation can serve the same function as fission. Some annelids fragment, while others divide by forming head segments midway down the body and then splitting into two new individuals. Many seaweeds and many corals may reproduce mainly by fragmentation. Tropical storms may benefit a staghorn coral population by dispersing newly created fragmented individuals. Colonial invertebrates may reproduce asexually by fission of whole individuals in the colony. As opposed to the production of new independent individuals, this process produces genetically identical and morphologically similar modules. Such colonial animals as encrusting sponges, coelenterates, and bryozoans utilize this mode. Marine algae and angiosperms include many vegetatively reproducing species. The marsh grass Spartina alterniflora usually consists of a large number of plants connected by a rhizome system. It can be demonstrated by biochemical marker techniques that genetically identical plants, all of which have derived from one progenitor, may cover tens of square meters. A marsh meadow is thus typically a series of clones. The value of vegetative spread varies with the biology of the individual group. In some corals and sponges, the overall form of the colony is important in the collection of food. In certain cases, the strength of the colony is helpful in resisting strong currents. Large colony size may also be an advantage. Larger colonies will have a greater ratio of living surface to periphery exposed to moving sediment. Therefore, they will be superior to small colonies in their ability to survive the movement of sediment along the bottom. For the same sort of reason, colonial forms may survive the attacks of predators, at least those attacking from the side. Colonies of the colonial Ascidian Bacillus schlosseri are usually founded by a single sexually produced larva, which attaches to a hard surface, metamorphoses, and then reproduces asexually. However, sometimes the larvae settle in aggregations. Using a unique genetic marker at an enzyme locus, Richard Grossberg and J.E.F. Quinn demonstrated that such aggregations consist of larvae that are genetically similar in tissue compatibility. These larvae can fuse and form larger initial colonies, which are more resistant to predation and, because the initiation of reproduction is size-dependent, may reproduce sexually at an earlier age. The phenomenon of immunological recognition of genetically related individuals seems to be widespread among groups that may benefit from group living. On eastern Pacific shores, the anemone Anthopleura elegantissima lives in large clones of several hundred to several thousand individuals, all derived from a single colonizing larva that repeatedly divides asexually. At any time, it is usually possible to catch some individuals in the middle of fission. Lisbeth Francis found that contacts between individuals of different clones result in a stereotype aggressive response. The affected anemones raise their tentacles and expose acrorhagi, which sting individuals from the other clone. No such aggression occurs between individuals within the same aggregation. This behavior can be interpreted as the defense of a communal territory, but it is not known what benefit is obtained from the defense, reproduction, demography, and life cycles. Natural selection on reproduction and life cycle age of first reproduction, reproductive effort, and longevity may evolve in response to different age-specific patterns of mortality and predictability of reproductive success in the population. Not all marine organisms spawn for the first time at the same age, nor do all devote the same proportion of available resources to reproduction. Consider the 5 species of Pacific salmon. All spend 3 years or so at sea while feeding as adults. They then make the long return trip to their spawning grounds, the tributary from which they originally came. During the trip upriver, they change morphology significantly, and by the time they spawn, they have no ability to return to the sea. They are so weak after spawning that they lie on the spawning beds and soon die. By contrast, Atlantic salmon spawns but then may go through at least one more migration, spawning cycle. Why should there be such a difference among species? Additional differences among species can be found in the age of first reproduction, proportion of energy devoted to reproduction, or reproductive effort, and whether the animal spawns only once or more than once. The theory explaining these differences is based partially on the premise that there is a cost of reproduction. We would expect evolution to maximize the total reproductive output over the lifetime of an organism. If there is a cost to reproduction, it then matters how reserves are allocated to reproduction versus somatic growth. If more reserves are devoted to reproduction, there will be fewer that can be devoted to growth. If mortality during reproduction is heavy, then any investment of resources into growth may be wasted because the organism will probably die before getting a chance to reproduce again. High adult mortality, therefore, selects for earlier reproduction. If adult mortality is low, it may pay to invest in additional growth to ensure repeated reproduction because larger adults can often produce more gametes and are often more experienced at winning mates than are smaller adults. Low adult mortality, therefore, selects for later age of first reproduction and for repeated reproduction. Environmental uncertainty is also an important factor and can be related to the potential success of a new year class. If environmental uncertainty is high, a new group of juveniles may not survive. This imposes a selective force for repeated reproduction to ensure that some year class of juveniles will carry on the population. In effect, a female engaged in such repeated reproduction is hedging bets against a future bad year. Table 7.1 compares age of first reproduction and reproductive span with variation in spawning success for 5 species. Fishes with relatively high spawning variability tend to have a longer reproductive span than those with low variability. The age of first reproduction, the degree of investment in gonads, and even longevity are all known to be genetically variable. It therefore is likely that natural populations are continually subjected to varying pressures of natural selection that act on this variation to change the life history traits of marine species. Some correlations we see in the ocean fit our expectations, but we would like a more direct approach to verify whether such changes are acting to cause evolutionary changes. Fishing causes natural selection on life histories. Fishing can be seen as an unplanned experiment that might alter life histories of natural populations very rapidly. Commercial fishing pressure should strongly influence the distribution of life histories of the exploited fish species. After all, species with low reproductive effort and late age of first reproduction will be the first to be seriously affected by intensive fishing. By contrast, species with early reproduction and high reproductive effort would be best suited to withstand the onslaught. It also stands to reason that life histories may evolve as a response to fishing. Fishers have inadvertently performed an experiment for us. The spiny dogfish Squalus acanthias is fished heavily in Europe but until recent years was hardly fished at all off the American Atlantic coast. As a result, spiny dogfish populations were far denser in American waters. The European populations have an earlier age of first reproduction and higher reproductive effort in the first spawning season. Assuming that all other things are equal, this would indicate that fishing pressure has selected for reproductive tactics that ensure more reproduction. Recent increased fishing pressure on North American populations will probably yield the same effect. In Chapter 21, we discuss an experimental approach that demonstrates strong life history responses of fish populations to simulated fishing, which may slow recovery of the populations once fishing is relaxed. Commercial fishers inadvertently performed an experiment that tested certain aspects of the theory explaining the size at which sex switching occurs. Species of the prawn genus Pandalus are fished widely in both the North Atlantic and Pacific Oceans. The prawns mature first as males but, after a variable period of time, switch to being females for the rest of their lives. A prawn fishery commenced in Danish and Swedish waters about 1930. Following the rapid increase of fishing pressure, the average body size of the catch decreased and the threshold size of switching to females also decreased. This development can be interpreted as follows: increased mortality will cause females to become relatively rare if males continue to switch sex only when they have reached a large body size. If a new genetic variant appears that switches at a smaller size, it will produce more offspring. Owing to natural selection for these variants, the size of sex switch will decline over time. If fishing pressure is very severe, it might pay for some individuals to mature first as females rather than go through a male stage. Such populations exist. Migration fishes, crustaceans, turtles, and marine mammals often migrate between spawning and feeding grounds. Many marine species have a reproduction and migration cycle as shown in Figure 7. 9. Juveniles drift or swim from a spawning area to a nursery area, and then move to an adult feeding area as they grow older. Adults then migrate back to the spawning area. The cycle of spawning, drift, and active migration back again reflects a maximization of reproductive success by feeding and spawning in different optimal sites. The movement from nursery grounds to different adult feeding grounds may reflect competition between juveniles and adults for limited food resources, or it may result from differing food requirements of the different age classes. In many cases, fish spawn for more than one reproductive season, so there is repeated migration between the adult stock area and the spawning area. Such migratory patterns are strongly timed with seasonal changes that strongly affect temperature and food availability. Spawning must be timed precisely to allow development of larvae in water conditions with favorable temperature for development and with seasonally abundant food supplies for feeding larvae. Marine turtles are a good example of the great difference in optimal breeding sites versus feeding sites. Females of all species lay and bury eggs in sandy beaches in the subtropics and tropics. The warm temperature within the sand ensures proper development, and the specific temperature even determines the sex of hatchlings. On the other hand, different turtle species migrate to feeding sites that depend on the food specialization of the turtle species. Green turtles usually feed on marine benthic plants and some attached bottom invertebrates and, therefore, migrate to sites where they are abundant. This may involve trips of hundreds or thousands of kilometers. Leatherback turtles feed on jellyfish. They too lay eggs on tropical beaches, and those that nest in the Caribbean migrate to cool high-latitude waters of Canada and Europe. Concentrations of leatherbacks can be found in association with continental shelf areas where jellyfish are usually abundant, which suggests that migration patterns and homing might be influenced by uneven prey distributions of jellyfish in the ocean. Migratory patterns involving estuaries are classified on the basis of the location of spawning and adult feeding grounds. Diadromous species divide their lives between residents within the freshwater parts of estuaries and in the open sea. Anadromous fishes spend most of their time in the sea but breed in fresh water. Catadromous fishes usually spend their adult lives in fresh water or tidal creeks but move to the sea to breed. Many species feed and breed near the oceanic coast or in the open sea, although they migrate between different localities. Migratory species vary in their degree of homing. Pacific salmon species are born in freshwater tributaries and migrate to the sea after a few months. They return 2-3 years later to the coastal area of their native river with the aid of detection of the Earth's magnetic field and then return to their native tributary using their olfactory system, spawning in the very small rivulet where their parents spawned. The timing of the return is crucial to allow favorable conditions for spawning. By contrast, the early larval stages of the herring Clupea harengus drift shoreward from the spawning grounds. As they grow, the herring move to deeper water and feed upon larger zooplankton. They return to a spawning ground after a year, but homing is not exact. The herring often occur in distinct stocks or separated populations that maintain separate migratory routes. The American eel Anguilla rostrata and the European eel A. anguilla undergo one of the world's most spectacular and enigmatic migrations. Both species migrate from freshwater rivers, ponds, and estuaries in Eastern American or European waters to spawn in partially overlapping areas in the Sargasso Sea in the middle of the subtropical North Atlantic Gyre. The trip to the spawning site is poorly known, but adults are believed to swim in deep water. After spawning and zygote formation, the larvae drift north and eastward in the Gulf Stream. American eels then move from a range of latitudes, westward across the continental shelf, toward shallow water Eastern American rivers and estuaries, whereas European eels continue to drift across the Atlantic and are carried by currents into shallow waters there. When the larvae reach an estuary, they metamorphose into the glass eel phase, which takes advantage of tidal behavior to move upstream by locating themselves up in the water during flood tide stage. The two species are distinctive in chromosome count, in various biochemical genetic markers, and even in the number of vertebrae. Other species of anguilla, such as the Australian A. Rhea and hardy, also undergo very long migrations that depend on currents to carry larvae back to adult habitats. It is not clear at all why these eel species migrate so far to their spawning grounds. Could these particular areas be optimal for spawning? If so, how were they discovered at such a great distance? The movement of larvae must be passive, so the further passage of European eels must be determined genetically. A fascinating population has been discovered in Iceland, which appears to include an excess of eels that are intermediate in morphology and genetic markers. The Icelandic population includes hybrids of A. rostrata and A. anguilla, which hybridize thousands of kilometers away in the Sargasso Sea. A recent study employing single Nucleotide polymorphisms from DNA sequencing that isolated species-specific alleles suggests that about 11% of Icelandic eels are hybrids, and some are even backcrosses between hybrids and either European or American eels. These hybrids appear to be migrating halfway across the Atlantic and winding up in Iceland. One wonders whether the migration cycles of the North Atlantic species started millions of years ago, when the Atlantic was narrower. Then, the opening of the Atlantic might have isolated new species on either side. Alas, the data do not support this idea. Menegishi and colleagues used mitochondrial DNA sequences to estimate the time of divergence of eels, based on the notion that greater DNA distance between groups would imply greater time since evolutionary divergence. The estimate of divergence between A. anguilla and A. rostrata is 5.8 million years, which is much younger than the age of divergence of the Atlantic. Green turtles are still something of an enigma to marine scientists. It is clear that they migrate hundreds to thousands of kilometers between feeding grounds and beaches where females lay eggs. Turtles that lay eggs at Tortuguero on the Atlantic coast of Costa Rica, for example, can be found at other times throughout the Caribbean, including Venezuela, Mexico, Cuba, and Puerto Rico. At least some tagged females return repeatedly to the same beach to lay eggs, figure 7. 12. It is unclear whether this behavior starts at birth, when hatchlings move to the sea, or whether females just follow others to good beaches to lay eggs. Right now, the evidence favors the hypothesis of imprinting at birth. Female turtles from different beaches are genetically distinct, but turtles from the same beach are very similar. This can be shown by studies of the DNA in their mitochondria, which are inherited maternally. Populations from different beaches have probably been isolated from each other long enough to diverge genetically. The mechanism of migration routes of sea turtles is a major question, even at first hatching, escape from the nest, and running to the sea. On small scales, sea turtles, when hatched, first respond to light and wave action in order to find the water. At larger scales, however, it appears that perception of the Earth's magnetic field is a means of providing a map for sea turtles that may swim thousands of kilometers. For example, young loggerhead turtles Caretta migrate from the waters of the southeastern United States clockwise around the North Atlantic and return back to American waters. Lohmann and Lohmann subjected turtle hatchlings to variation in magnetic fields and demonstrated that they can respond to variation in both the intensity and the direction of a magnetic field. Both of these factors vary with latitude, which means that sea turtles have the means to map their location and respond accordingly. For example, in the laboratory, loggerhead turtle hatchlings orient toward the east in a field magnetic intensity that corresponds to being along the southeastern United States coast. This would put them in the Gulf Stream. But when they are subjected to a magnetic field intensity corresponding to the eastern Atlantic, they orient toward the west, which in the Atlantic would keep them within the North Atlantic gyre and move them toward the tropics and eventually back to the tropical western Atlantic. Anadromous fish species are more common in high latitudes, and catadromous species dominate low latitudes. This pattern may be related to the availability of adult food. Fish migration is one of the most perplexing phenomena in marine biology. Some species breed in fresh water but migrate, sometimes thousands of miles, to feed in salt water. Others do the reverse. Why should some species move in one direction, whereas others take the opposite tack? Migration should make sense as a way to increase the growth rate of a fish population. After all, if migration had costliness as its only characteristic, genetic variants that did not migrate would win out in the evolutionary race. And indeed, migration costs a lot. Such a journey increases the risks of starvation, predation, and simply becoming lost. It must be that migration produces increased benefits in terms of increased reproduction at a suitable spawning ground and increased growth in a good feeding area. As it turns out, there is a systematic variation in the relative abundance of anadromous and catadromous fish species in different regions. Anadromous fishes are more common in high latitudes, whereas catadromous fishes seem to dominate tropical habitats. Despite some glaring exceptions, the Atlantic eels are catadromous but live in temperate and boreal North Atlantic latitudes. Overall, the pattern holds well, at least for the northern hemisphere. Zoologist Mart R. Gross and colleagues discovered an important key to success in migration. Food abundance is obviously important for fish survival and growth. As it turns out, high-latitude rivers and streams have lower overall productivity, and therefore less fish food, than the adjacent oceans. Thus, anadromous fish spend their time feeding in the ocean but come into freshwater bodies briefly to spawn. By contrast, tropical oceans are very low in productivity. But tropical rivers and streams are very productive. This suggests that the difference in migration is not really paradoxical. Fishes are feeding where the food is. An interesting experiment confirms the hypothesis. The Arctic char is anadromous, but experimental additions of food to fresh waters decreased the migrations back to the sea. McDowell has argued that the relationship holds only in the northern hemisphere, where anadromous fish diversity is greatest in the range of 50-60 degrees north latitude. But there is no corresponding peak in the southern hemisphere. He suggests that after the continental glaciers retreated 8,000-10,000 years ago, a large number of previously anadromous species just reinvaded these northern latitude rivers as they reopened. Larval dispersal, the long and the short haul modes of dispersal marine invertebrate. Offspring may be brooded or released as small adults, dispersed usually short distances by means of relatively short-lived, yolk-dependent lithotrophic larvae, or dispersed great distances by means of longer-lived, plankton-feeding planktotrophic larvae. The hallmark of larval production and dispersal is a complex life cycle. An adult stage gives rise to a dispersing larva, which moves to a new site and completes the cycle by establishing itself and eventually growing to reproductive maturity. Figure 7.13 shows an example of such a cycle. Although there is a continuum of dispersal distances, several qualitatively different types of release and spread of larvae result in modes of dispersal distances. Direct release of individuals next to adults is the shortest type of dispersal. Many species are viviparous, that is, they bear live embryos and then release juveniles as crawling miniature adults. Such young may crawl directly onto the mud or rocks. The Atlantic periwinkle Littorina saxatilis broods fertilized eggs in a modified oviduct, and fully shelled young snails are later released. Many Antarctic species of featherstar develop their eggs in brood chambers located near the gonads in the arms at the pinnal bases. Other species are oviparous, producers of young that hatch from egg cases. Several species of the drilling genus Nacella lay egg capsules that they attach to rocks. Embryos develop in the eggs, then hatch and crawl away as fully shelled juveniles. It is extremely common for myofauna to have direct release, since any sort of current-driven dispersal will likely take them to inappropriate habitats. The small size of myofauna also restricts them to releasing very few young, which puts a premium on maximizing survival. Most invertebrates have larvae that swim for varying amounts of time before settlement and metamorphosis. Lithotrophic larvae are swimming larvae that depend on nourishment from the yolk provided in a relatively large egg. The larvae have no feeding or digestive structures. Capable of limited swimming, lithotrophic larvae of most marine species spend a few hours to a day or so moving either along the bottom or up in the water column. The development time of this mode of larval development cannot permit dispersal for usually more than hundreds of meters. Lithotrophic larval development occurs in species of many phyla, including mollusks, annelids, and ectoprocs. An interesting exception is echinoderms, whose high-latitude species commonly have lithotrophic larvae that remain in the plankton for weeks, which is unusual for this type of dispersal. Planktotrophic larvae feed while they are in the plankton. They usually have specialized larval feeding structures and digestive systems. They feed on planktonic bacteria, algae, and smaller zooplankton and usually drift for 1 to several weeks in the water column. Because of the time they spend in open waters, they can disperse over long distances. Development often proceeds through a number of planktonic stages, when the larva is competent to settle and metamorphose on the bottom. If a suitable adult substratum is not found, the larva may be capable of delaying metamorphosis. Metamorphosis often involves a major reorganization of morphology from the larval to the adult form. While closely related species with planktonic larvae may have either planktotrophic or lithotrophic development, it is quite rare for a single species to have the ability to produce both larval types. Species with more than one larval developmental mode are called poecilogonic and are found in a few groups, including polychaete annelids, sea slugs, and a cynogastropod. For example, Lisa Levin found that the small mud-dwelling polychaete Strablospio benedicti is widespread on North American mudflats and has either planktotrophic or leucothotrophic larvae, depending on the local population. A southern Californian species of the Sacoglossum sea slugalderia also shows poecilogony and tends to produce leucothotrophic larvae in summer and planktotrophic larvae in winter. The seasonal switching may be due to the presence of planktonic food, but the explanation is still open. Leucothotrophic larvae are often covered with ciliary bands, which are used for locomotion. However, planktotrophic larvae use ciliary bands or tufts to move and to feed upon the planktonic food. Figure 7.17. The small size of the cilia and the low velocities of ciliary movement cause planktonic larval feeding to operate at low Reynolds number. This means that viscosity dominates inertial forces in fluid behavior. The cilia must beat to propel water and food particles. Although there is a great deal of variation in morphology, a surprising number of taxonomic groups of organisms have ciliated bands. The cilia beat and create a current across the band. To capture a particle, it appears that a cilium suddenly reverses its rowing motion toward downstream. The reverse beat captures the particle and traps it upstream of the ciliary band. The ciliated bands are also generally used to propel the larva through the water, which exposes the larva to new micropatches of food. It is likely that although the same cilia serve for propulsion and feeding, the ciliary action for the two functions involves trade-offs. Capture of algae involves adding small vortices by ciliary reversal. But these vortices are added at the expense of larger propulsion of the larvae. Ciliary propulsion in sea star larvae involves the production of large vortices around the swimming larvae, but feeding may involve a different regime of ciliary control. The larva of the blue mussel Mytilus adulis, which lives on intertidal and shallow subtidal rocks and cobbles, is a good example of a typical planktotrophic larva. Sexes are separate, and the animals release eggs and sperm into the water. Within 9 hours after fertilization, the larvae are completely ciliated and are strong swimmers. Within 5-7 days, fully developed feeding veliger larvae develop. Normally, larval life may be 4-5 weeks. By contrast, the Atlantic mud snail Tritia obsoleta produces fertilized eggs laid in cases that are attached to rocks. Larvae emerge from the cases and swim away. Planktotrophic larvae may also be released directly from the mother, as in the acorn barnacles and many species of crab. Larvae of bony fishes are obviously different from the foregoing descriptions of invertebrate larvae. Most marine fish species spawn buoyant eggs and sperm into the water. Hatched embryos first develop into larvae with a prominent yolk sac. Once the sac is absorbed, the larvae usually feed on zooplankton. Fish larvae remain in the upper water column from 2 weeks to as much as several months. They then metamorphose into adults, with adult organs. The degree of morphological change during metamorphosis varies from very little to a major reorganization, as when the American and European eel Leptocephalus larvae metamorphose into elvers. Coral reef fish larvae are notable for their rapid swimming speeds, which may be related to a need for reaching more specific final habitats. Reproductive timing and egg-size gamete production. And larval life must often be timed precisely to allow settlement and promote dispersal, to avoid being swept to inappropriate habitats, and to counter predation. The commitment of either gametes or larvae to the water column entails great dangers of extensive and possibly total mortality. We discussed earlier the problems associated with ensuring that fertilization is maximized. In estuaries, larvae in the surface low salinity layer may be lost to sea. Predation on gametes and larvae also can strongly depress reproductive success. These dangers require precise timing in many cases to avoid gamete and larval death. For example, animals living or breeding in the highest part of the intertidal zone must have precise larval release schedules because the animals may be inundated only once every 2 weeks during spring high tides. The Atlantic marsh snail Melampus bidentatus lives in the highest part of the tide zone and breathes air, but it lays eggs that develop into swimming larvae. These larvae are launched at a spring high tide and return to settle during the next spring high tide. The California grunion Lurestes tenuis comes onto sandflats to spawn at spring high tide at night. A female rides the swash onto the beach accompanied by several males. She twists her body, digs a pit in the sand, and lays eggs at the highest level of the tide, while males curl around the female and deposit sperm, which fertilizes the eggs as they are laid. Males and females are washed down the beach slope to the water and swim away. For about 2 weeks, the high tides fail to reach the level of the beach where the eggs were buried, but the next spring high tide washes out the embryos, which detect the cue provided by wetting and hatch into swimming larvae. Tidal environments are most challenging because gametes and larvae may be trapped in the surface layer during the day, when visual predators are active. Therefore, one might expect strong selection for larvae to be moving in the surface waters at night. This has been found in the coral reef flat damselfish Pomocentris flavicata, which lays eggs at dawn. Its embryos hatch as larvae on the fourth sunset. Egg production is timed so that hatching occurs at the time of the month when sunset coincides with ebb tide, which at once lowers predation and sweeps the larvae offshore. Many intertidal crab species are exquisitely timed to release larvae at high tide. This release must be coordinated with copulation and embryo development. Larval release itself must be coordinated with tidal amplitude, tidal cycles, and light-dark cycles. Ideally, larvae should be released during highest tide and at night to maximize larval release, but also to be in the dark to avoid predators. Species are often capable of releasing larvae in different rhythms, depending on variation in tidal cycles. As mentioned in Chapter 2, tidal cycles can vary considerably in different locations. For a number of crab species, Stephen Morgan and John Christie found that crabs were able to adjust to radically different tidal cycles on the Pacific and Caribbean sides of Panama. In nearly all cases, females released larvae at the time that high tide and dusk plus 1 hour coincided. It is of interest that there are some tidal regimes where this is not possible, as within an estuary. Fiddler crabs release larvae in time with the tides, but the larvae also have an endogenous rhythm that allows them to rise up in the water column in an estuary when the tide is going out. They drop to the bottom when the tide rises. This allows them to efficiently move out to sea and not get swept up estuary when the tide is rising. Variation in egg size is considerable among marine species, and this may relate to different consequences in terms of mortality. Although variation in age of reproduction is considerable, equally noticeable is the great variation in egg size in marine animals, sometimes between closely related species. One expects larger eggs to have more yolk nutrients, but this is only broadly true, and there are exceptions. In large measure, the variation remains unexplained, but there are some general patterns. As mentioned earlier, lessitotrophic larvae develop from eggs that are much larger than planktotrophic eggs. Eggs of directly released juveniles are largest of all. As a general rule, planktotrophic eggs produced per female are also much more numerous than lessitotrophic eggs, which are, in turn, more numerous than the eggs of directly releasing species. This suggests a strategy that, in turn, may determine egg size. Planktotrophic larvae swim in the water far longer and probably have much higher mortality rates than do lessitotrophic larvae, which are in the water for only a few hours. In turn, directly releasing species deposit their young in appropriate habitats, thus further minimizing mortality. It seems likely then that the female, by producing a large number of planktotrophic eggs, hedges her bet because far fewer young will survive relative to lessitotrophic eggs. Some have suggested that the strategy of producing an intermediate-size egg with intermediate numbers may offer the worst of both worlds, producing high mortality with relatively low numbers of eggs and young. Thus, we expect to observe either the low-number quality care approach or the high-number leave-them-alone approach. The interested student will find that the subject of egg size is quite controversial. The microscale, larval settlement, metamorphosis, and early juvenile. Survival planktonic larval life is a transition from pre-settlement morphology and behavior to competence for settlement and metamorphosis. Planktonic larvae develop from the zygote into a free-living organism that develops through a series of stages adapted to remain in the water column to stages involved in settlement and metamorphosis. The stage at which a larva can touch the seabed and metamorphose into a benthic adult is the time of competence. A competent larva faces the important choice of settling and metamorphosing on the best adult substratum possible. But what if an acorn barnacle larva encounters only mud? In some species, delay of metamorphosis is possible, but in others the larva is programmed to settle and metamorphose at a specific time, with no scope for delay. Morphology and behavior may change during the time of planktonic larval life. For example, planktotrophic larvae of the eastern oyster Crassostrea virginica spend about 2 weeks in the upper water column, but move more slowly and spend time in bottom waters in the 3rd week as the larva approaches metamorphosis. During the 3rd week, the larva develops a foot, which can probe the bottom for suitable habitat to metamorphose. In the 3rd week, the larva is competent to settle and will be especially stimulated to settle and metamorphose on the shells of adult oysters. We have an incomplete understanding of the internal signaling that leads from larvae remaining as feeding individuals in the water column to finally transitioning to a stage of competence to settle and metamorphose. In a wide variety of distantly related phyla, high activity of the nitric oxide pathway interacting with heat shock protein 90 contribute to forestall the tendency to metamorphose. NO production relies on the enzyme nitrogen oxide synthetase and serves as a signal to regulate larval development. As larval life proceeds, the activity of NO declines. Signals that suppress NO or HSP90 lead to metamorphosis. Details have been worked out in a few species, but external environmental signals must interact with the NOS HSP90 complex and other pathways to lead to larval competency and settlement and metamorphosis. See example. Castellano et al., 2014, but also see Ueda and Degnan, 2014. Planktonic larvae use light and pressure cues to maintain depth for optimal feeding and location of settling sites. Planktonic larvae reach a stage at which they must locate the adult habitat, where they will settle and attach before metamorphosis. At the stage of competence, larvae of some species can delay metamorphosis for some time, but other species appear to have a biological clock for settlement within a specified time and will die otherwise. Although the vast majority of larvae die before reaching a potential adult habitat, the road ahead is still dangerous for the survivors. Natural selection and evolution favor any larval features that enhance location and settlement on the proper substratum. Figure 7.19 provides a general outline of the stages of larval selection. In the plankton, larvae pass through one or more stages of photopositive and photonegative behavior. These permit the larvae to remain near the sea surface to feed and then to drop to the bottom and settle on the proper habitat. Larvae of intertidal animals are photopositive during their entire larval life so that they may feed in the surface waters and settle in the intertidal zone. Richard Grossberg examined the water column distribution of intertidal barnacle species in California and reported that the highest intertidal species was to be found at the surface during larval life. Species living in the mid-intertidal, however, were found a meter or two below the surface. Intertidal larvae are apparently capable of subtle adaptations to maintain the proper tidal height at the time of settlement. Subtidal species, by contrast, are first positively phototactic but then switch to negative phototaxis just before the time of settling. Larvae are also capable of responding to pressure differences that might enable them to select a specific depth during the period of feeding, dispersal, and settlement. Pressure is a good indicator of water depth. Planktonic larvae must maintain their proper depth in the water column during the dispersal phase either to feed on phytoplankton that are abundant near the surface or simply to avoid sinking before development is completed. Most larvae have slightly negative buoyancy, but simple turbulence retards sinking. Larvae can be simple and ciliated, but planktotrophic larvae usually have a swimming organ. Young postlarval mussels secrete monofilamental byssal threads more than 100 times longer than the larval shell. The filaments exert viscous drag force sufficient to reduce greatly the sinking rate, thereby enhancing dispersal. Even if turbulent mixing prohibits the sort of vertical stratification observed by Grossberg in larvae, there is good evidence that larval selectivity strongly influences where settlement occurs. Jenkins studied settlement of planktotrophic larvae of 2 species of the barnacle genus Thamylus in waters of the United Kingdom. While larvae of both species were thoroughly mixed in the water column, settlement patterns of each of the two species were quite specific and corresponded to the different environmental occurrences of familous species, one of whose adults were found in exposed habitats and the other in more protected habitats. Larval supply may, therefore, be important in total abundance of settlers but not in determining the microhabitat where they wind up. Larval behavior is clearly important in habitat selection at the microscale. Amazingly enough, deep-sea hot vent environments have species with planktotrophic larvae. Hot vents are scattered throughout the deep-sea floor and are ephemeral because the hot nutrient-rich sources give out after a time. Dispersal might thus be expected. The protoconch size of hot vent bivalve mollusks is of the typical small size for planktotrophic larvae, and isotope data from the larval shell show that the larvae spend a time in water that is warm, like surface oceanic water and unlike the deep sea. Vent bivalve larvae must make the seemingly impossible trip to the surface waters, and then some manage to locate an incredibly rare appropriate deep bottom environment. By contrast, archaeogastropods near the vents have large yolky eggs and probably have larvae with quite limited dispersal. It may be that these eggs hatch as swimming but non-feeding larvae, with strong dispersal abilities, relative to their non-vent relatives. At the scale of meters to centimeters, larvae use chemical and biological cues for a final settlement site. After touching the bottom, larvae must be able to determine whether the substratum is suitable for adult existence. Larvae use chemical cues and mechanical cues to detect suitable settling sites. Almost all larvae prefer surfaces coated with bacteria. Sterilized sand or rock usually inhibits larval settling. Larvae of some sand-dwelling polychaetes can select sand grains of the appropriate size, whereas rock-dwelling barnacles have larvae that move preferentially to surfaces with pits and grooves, which provide a secure attachment against wave action, predators, and competing barnacles that might overgrow or undercut them. Although barnacles are often found in cracks, it is often not clear whether this is the result of preferential settlement or differential survival after settlement. An important physical cue is the local hydrodynamic environment. Many bivalves, for example, cannot suspension feed efficiently in high flow, but zero flow is also bad because no current means no phytoplankton delivery. The final settling stage larvae of the barnacle Balanus improvisus encounter substrates and actively evaluate the current speeds at different microsites. If water movement exceeds 5-10 cm S1, the larvae actively reject the site and move on to sites where currents are less. This selection relates directly to the pore efficiency at which the adult barnacles will be able to feed at the higher current speeds. In gregarious settling, larvae settle on adults of their own species. This usually requires direct contact of larvae with adults because the chemical cue is a relatively insoluble molecule, such as arthropodin in barnacles. Zimmerfaust and Tombury discovered that the cue for settling of oyster larvae is a soluble peptide. Such a waterborne settling cue exists in effective concentrations only within a few millimeters of the bottom, but larvae sinking within the bottom boundary layer can detect this cue and settle. The greatest concentration of such a settling cue would likely emanate from the water coming from adult oysters, which would concentrate settling of oyster larvae on adults of their own species. Larvae are often entrained in currents too strong to swim against, but they frequently drop down toward the substratum and make arc-like swimming patterns. On encountering the cue, they can slow down, settle, and metamorphose. Field experiments show that oyster larvae actively settle on artificial substrates that leak such peptides into the water. Gregarious settling allows a larva to settle in a site where adults have already settled and survived. There is the disadvantage, however, of being eaten by adults of the same species. There also is the problem of how, in a gregarious species, a pioneer can ever manage to colonize a new site. Tunan and Pollock discovered that the colonial tube-dwelling polychaete worm Hygroides dianthus has two distinct types of planktonic larvae. One seeks adults of its own species, and the other is specifically a pioneer, a larva that seeks new, bare, hard substratum. The cue for gregarious settlement is a waterborne compound emanating from benthic adults. The proportion of larvae in a given spawning that adopts one of the two modes is a highly inheritable trait, which suggests that the two-strategy approach is under strong natural selection. Planktonic larvae may also use the chemical characteristics of other benthic species as cues. For example, adults of the hydroid family Proboscidactylide live on the tubes of members of the polychaete annelid family Cybelidae. At settling time, the larvae can detect and settle on the tentacles of the worm. After settling, they move down the tentacles and live as adults on the tube. The mechanism probably involves chemical attraction. Some bryozoan larvae are attracted to seaweeds, and organic substances can be extracted from some seaweeds that will attract larvae onto another non-living surface. In other species, mechanical attraction is involved. Planktotrophic larvae of the mussel Mytilus adulis are attracted to filamentous red algae, but the larvae will also settle on fibrous rope that has approximately the same texture. This attraction is used to great advantage in mussel mariculture. After settling, larvae may move a short distance, no more than a few centimeters, to a better site. This is very important for larvae of sessile species, whose movements at this point commit them to life at one location. Newly settled barnacle larvae can move to a small degree, locate optimal microsites, and space themselves away from other barnacles. The tubeworm Spirorbes borealis settles randomly on the seaweed Fucus serratus, but individuals then crawl and space themselves evenly. Avoidance of crowding reduces overgrowth and competition for food. In barnacles, among which settlement is often very dense, the reduction of crowding that follows a period of mortality after settlement greatly enhances the subsequent expectation of life. When they reach the substratum, marine planktonic larvae encounter a complex physical and chemical environment. As we have discussed, larvae may select settling sites based upon chemical cues. However, there are probably a number of chemical cues that may lead to avoidance of a local site. Unfortunately, we know very little about these cues, even though they may be a major means of local site selection. Sarah and Wooden and colleagues demonstrated that noxious chemicals released by benthic animals may reside in the sediment-poor waters and can inhibit settlement by larvae. In soft sediment intertidal flats of Washington, the infaunal terabellid polychaete Thelopus crispus is locally abundant and harbors toxic bromine-containing aromatic compounds. These compounds are found in the sediment-poor waters adjacent to the worm. Such sediments are actively rejected by settling larvae of the Nereid. Polychaete Nereus vexillosa, a common resident of sandy shores in the area. This result suggests that negative chemical interactions may also be important in determining the settling and metamorphosis of planktonic larvae. Finally, a larva commits itself and metamorphoses. In the case of barnacles, for example, the football-shaped cypress develops adult appendages, and a basal plate is laid down. The energetic cost of metamorphosis often is so severe that the animal must feed immediately afterward. Some sea star species die if they fail to find a prey item within a couple of days after metamorphosis. If larvae of the place Pleuronectes platessa are starved of zooplankton for more than 8 days, a point of no return is reached, and they will not have the energy to feed subsequently. Older larvae can survive at least 25 days before the point of no return is reached. It may be that early larval development in these fish is also very costly. The mesoscale, transport by currents, loss of larvae, and retention mechanisms, planktonic dispersal. Success of marine species is strongly controlled by currents that transport larvae and may have important ecological consequences. Planktonic dispersal does not guarantee the ability to survive a long trip across the ocean or to colonize new habitats successfully. The overwhelming majority of planktonic larvae die in the water column from predation or starvation, or are swept to inappropriate habitats. Larvae of rocky shore invertebrates are released in currents and may easily be swept to inappropriate habitats. Along the southern New England coast, a longshore current would tend to sweep larvae from the rocky coasts of Rhode Island to the sandy south shore of Long Island, New York, and most larvae of the rocky shore species would be doomed. Species with non-planktonic larvae dominate the Galapagos Islands, off Ecuador, where currents carry surface waters offshore for a large portion of the year. As a result, species with planktonic larvae are relatively rare in the Galapagos. Most recruits, even of planktonic larvae, probably come from nearby. Figure 7.21 shows the general possibilities of larval transport. Depending on the local current regime, larvae might be swept offshore, or they might even be brought on shore by wave trains, internal waves that move material toward shore. Longshore drift is common, and larvae usually are moved parallel to coastlines to some degree. This may permit rapid extension of a species' biogeographic range. Finally, inhomogeneities in the coastline often create eddies, which may concentrate planktonic larvae. Although average dispersal distance per individual planktotrophic larva is probably on the scale of 10 kilometers, the potential exists for longer distance dispersal in a significant fraction of the planktotrophic larval cohort, and this potential must increase with the planktonic larval lifespan and with the favorable nature of ecological opportunities at the distant site. Rudolf Skeltima discovered that many planktonic larvae of coastal invertebrate species live in the open sea, particularly in major transoceanic surface currents. He termed such larvae teleplanic larvae and found transoceanic similarities in species whose larvae were common in the open ocean. Another particularly interesting case is that of the larvae of the coral Passalopora damicornis, which is widespread and dominant throughout Pacific coral reefs. Robert Richmond has shown that these larvae can live in the plankton and are competent to settle and metamorphose for periods greater than 100 days. This may explain the coral's broad geographic distribution. The larva's symbiotic zooxanthellae photosynthesize and provide food, which fuels the larva's journey over long distances across the open sea. Most larval exchange probably occurs along coasts, and strong coastal currents parallel to shore may increase the interchange of populations from various locations along the coast. The degree of interchange is known as connectivity. Connectivity can be strong between populations of a species if larvae typically move great distances along coastlines and are periodically reversed in direction. Alternatively, as we have mentioned, some circulation patterns cause larvae to be retained within local areas, causing the local population to be self-seeding. Patterns of connectivity can vary from region to region, and even from season to season, since strong coastal currents might move larvae in one season, but not in another. If some ecological opening exists, arriving larvae may flourish, reproduce, and continue to extend the species' geographic range along a coastline. Such invasions have happened several times during the last century, and the sudden expansions may have been aided by commercial shipping. In the mid-19th century, the shore periwinkle Littorina littoria was first noticed on the shores of Nova Scotia and spread southward. It had arrived at Cape Cod, Massachusetts, by the turn of the century and has now reached the Middle Atlantic states. It is unlikely that the species will spread much farther southward, given its thermal and geographic ranges in Europe. Many invasive species also have spread rapidly along a new coastline. Once having arrived despite the potential for dispersal, planktonic larvae often come to settle quite near their origin, owing to cyclonic or returning currents. Given the long lives of many planktotrophic larvae, one expects that dispersal should occur over great distances. A larva with a 30-day lifespan and a steady and straight current of 2 centimeters should disperse over 50 kilometers. This would suggest extensive mixing of marine species with planktotrophic larvae. The assumption of straight and steady currents is the problem with such a prediction. Not all currents move steadily or in one direction. Shapes of basins and large obstructions to flow, such as islands, often create eddies that cause larvae to travel in cyclonic flow patterns, sometimes to their parent populations. Paul Samarco monitored larval settling in the vicinity of an isolated patch reef in the Great Barrier Reef of Australia and demonstrated surprisingly reduced dispersal of planktonic coral larvae. Settling plates were placed at various distances from the patch reef, and a detailed study of local water currents allowed a prediction of sites where larvae might be concentrated. Settling occurred mainly in the close vicinity of the patch reef, suggesting that most larvae come from within the patch system itself. Settlement was greatest in a vortex of currents, where water was trapped and recirculated for a while before being swept to sea. A similar story emerges from the dispersal and current patterns associated with a population of the European lobster Nephrops norvegicus in the western Irish Sea. Alan Hill and colleagues discovered a cyclonic flow in spring and summer. This flow lies above a large patch of mud, which has the main population of the lobster in this region. In spring, newly hatched larvae spend approximately 50 days in the water column, and successful recruitment depends upon finding the mud patch. Larval distributions show a strong correspondence to a stratified water body enclosed by the small gyre system. This ensures that the larvae will settle in the vicinity of the mud patch. Currents may cause net movement toward the shore. General eastward currents and winds tend to keep eastern Atlantic larvae pressed against the shorelines of the Atlantic coast of Europe. Off most open ocean coasts, one can observe surface slicks that contain jetsam that seems to move toward shore. Surface floats off the California coast are often carried 1-2 kilometers shoreward in 2-3 hours. Planktonic invertebrate and fish larvae are 6-40 times more concentrated in these slicks than in the surface waters between the slicks. The slicks are formed by the interaction with the sea surface of tidally driven internal waves. Planktonic larvae may therefore be trapped against the shoreline. In recent years, episodic recruitment has been discovered to be the rule for settling megalopa larvae of the blue crab Calanectes sapidus, both on the east and Gulf coasts of North America. In some cases, these recruitment events last only a day and may be simultaneous over several hundred kilometers of coastline. Recruitment in the mid-Atlantic states region appears to be unrelated to internal waves but is correlated with onshore transport caused by wind stress and by spring flood tides, although other factors may also be important. Many coastal species have evolved mechanisms for retention of larvae near suitable adult sites, take advantage of current systems to return to suitable sites, or even benefit from seasonal reversals of flow that allow them to move out to sea and then return to suitable coastal adult habitats. Many coastal species have apparently taken advantage of persistent currents, tidal reversals, and even seasonal changes to reduce loss of planktonic larvae to unsuitable sites. Many estuarine species spawn within an estuary, but larvae and juveniles may spend some period of time in coastal waters, perhaps to avoid predation within the estuaries, which can be intense because of large predatory fish populations. Because they are entirely dependent on currents for transport, they must take advantage of the various wind and tide-driven transport sources discussed earlier to return to the estuary from adjacent coastal waters. As was discussed in Chapter 2, surface water flows seaward in moderately stratified estuaries, whereas the more saline bottom layer moves landward. If estuarine planktonic larvae held their position near the surface, they would be carried out to sea. This is a strategy employed by a number of species, which brings their larvae out into continental shelf waters. They may return to the estuaries after a larval life in coastal waters, and larval recruitment depends very strongly on often quite irregular onshore winds. But many other estuarine species avoid this seaward surface flow. Larvae of the eastern oyster studied in the James River Estuary, Virginia, differ in depth distribution from inert particles and are found in surface waters more frequently at the time of flood tide, which would tend to move them upstream. The larvae use the halicline as a cue for vertical motion, rising with the flood and sinking at the time of ebbing tide. Larvae of the mud crab. Rithropanopius horizii move upward on the rising tide and downward on the ebbing tide and stay near the bottom, which gradually concentrates the larvae farther up the estuary. The evidence suggests therefore that there are two alternative strategies for planktonic larval habits of estuarine species. Some species have larvae with adaptations for estuarine retention, rising on the flood and moving to the bottom during ebb tide. A consequence may be increased predation by large populations of smaller planktivorous fishes in the estuary. Alternatively, larvae may rise on the ebb to move to coastal waters and then depend upon wind-driven currents to recruit back to the estuary. While such larvae would be living in waters of lower predation, the strategy is plagued by a higher probability that the larvae will never return to the estuary. The water column is filled with predators that cause considerable planktonic larval mortality. In temperate waters, planktivorous fishes and ctenophores are important grazers in the spring and summer. Ctenophores are especially abundant in the inner shelf waters and are major predators of zooplankton. Predation is a major problem for larvae that are adapted to be retained in estuaries. Stephen Morgan has shown that 3 species of crustacea retained in a North Carolina estuary have evolved swimming larvae with pronounced spines. The mud crab Rithropanopius horizii, for example, has movable spines, which are erected when fishes come close. Other crustacean species whose larvae spend time in coastal waters experience less predation and have less completely developed spination. The life history of the blue crab demonstrates the precarious situation of species that export their larvae from estuaries onto the continental shelf. The work of Charles Epifanio and colleagues on the biology and coastal oceanography of the blue crab Calonectes sapidus in Delaware Bay shows just how fragile a marine planktotrophic life cycle can be. As mentioned previously, adults copulate within the estuary. Larvae are released at the time of high tide, and the early naupliar larval stages move out to sea on the ebbing tide. The movement offshore occurs in the early summer, figure 7. 29, when low-density and low-salinity water is moving from northeastern United States estuaries onto the continental shelf. Although most flow from this low-salinity water is toward the south, a northward wind combined with the Coriolis effect pushes some water eastward, which carries some of the larvae out on the shelf, just seaward of the estuary from which they came. It is this fraction of larvae that makes it back to the estuary. In the late summer and early fall, winds move southward and combine with Coriolis-driven Ekman transport to bring later-stage megalopa larvae back westward across the shelf to the coastal estuaries. This whole process probably results in a loss of over 95% of the larvae that were originally exported from the estuary, but apparently enough return to sustain the population of Delaware Bay. What is amazing is how complex the relationship can be between larval transport and physical oceanography. Once the final megalopa stage larvae arrive in the estuary, they rise in the flood tide to be washed inward and retreat to the bottom on the falling tide to avoid being washed out to sea. The two alternative estuarine strategies, estuarine retention and larval export to the continental shelf, likely have strong consequences for connectivity among populations and evolutionary divergence. In species with strong retention within estuaries, populations along the coast are probably strongly isolated from each other. But in species where larvae are exported to the shelf, larval exchange along a coast greatly increases connectivity. Species with the retention strategy would be predicted to show strong genetic differences among estuaries along the coast because isolation is strong and individual estuaries may have differing environments. On the other hand, local extinction of retained species within estuaries may be frequent and recolonized by sporadic immigrants from other estuaries along the coast. Coral reef fish usually have planktonic larvae of several weeks' duration, and one might expect similarly a great deal of larval loss to sea by currents. But instead, many species show strong evidence of adaptations to take advantage of currents to return to localized reef areas of their parents. It may be that adaptations for return are crucial, since coral reefs are literally islands surrounded often by inhospitable soft sediments. Many coral reef adults are associated with very specialized microhabitats. Clownfish species, for example, often live in association with the tentacles of sea anemones. Butterfly fish feed on the polyps of corals but also must live close to sites where they can rapidly swim to crevices for shelter from predators. Still, larvae swim in the open sea, often spending several weeks in currents feeding on small zooplankton. Are they randomly mixed throughout hundreds of miles of coral reef tracts? The evidence suggests otherwise. Almany and colleagues studied a butterfly fish on a marine reserve in Papua New Guinea, and labeled females by injecting them with an isotope of barium that is rare in nature, relative to the common 135 Ba. The isotope was translocated to eggs, and the larvae also had identifiable amounts of 137 Ba. The vagabond butterfly fish had larvae that spent about 38 days in the plankton, which might lead to the expectation that most would be lost from the small island reserve of 0.3 kilometers. But instead, about 60% of the butterfly fish returned to their native reef. Similar results were found for a shorter dispersing species of clownfish. The work of Robert Cowan and colleagues provides a great deal of insight into the mechanisms and implications of the homing ability of planktonic larvae of many coral reef species. The bicolored damselfish Stygastes spartitis is common in the Caribbean island of Barbados, and larvae enter the water column after egg hatching and remain for about 30 days until settlement. Paris and Cowan followed larvae in the ocean by means of closely spaced samples and found that larvae tended to stay in discrete patches for many days. Early larvae were found in surface waters, but later stage larvae descended to depths where currents carried them back to the patch reefs from which they originated. These results demonstrate that active behaviors facilitate homing and are analogous to those mentioned previously for retention of oyster and mud crab larvae in northeast United States estuaries. Larval retention must also work on the very large scale of upwelling systems, such as are found of the Pacific coasts of North and South America. Upwelling during La Niña periods tends to move planktonic larvae of benthic species in surface waters out to sea, where adult habitats will never be found. Some evidence in recent years demonstrates that some planktonic larvae can descend to great depths offshore and return back appropriate adult coastal shore habitats in coastward moving countercurrents. Vertical migration by crab larvae often results in a net landward movement within such upwelling systems. Planktonic larvae getting through major obstacles to the final destination. Planktonic larval populations may fail because of a lack of planktonic larval food or because of concentrations of toxic algae. Although primary production is often sufficient to feed larval populations, shortages or failures of phytoplankton can devastate a planktonic feeding larval population that might be produced only once a year for a brief period. Harold Barnes encountered a bad phytoplankton year in British waters in 1951. Early larval stages of barnacles could be found at first, but these soon disappeared because of starvation. It would be best for larvae if their release could be coupled with the spring phytoplankton bloom. Such coupling has been found in sea urchins and mussels living in the St. Lawrence estuary of eastern Canada, which spawn in response to high concentrations of phytoplankton. The response is similar when urchins and mussels are exposed to only the filtered water in which the phytoplankton lived. The response is also advantageous because zooplankton predators tend to be rare at the peak of the phytoplankton bloom, so larvae can eat and not be eaten. There may be cases in which larvae do not suffer, especially from food shortages. Echinoderm larvae may survive well despite an absence of phytoplankton, and many bivalve mollusk larvae are also resistant to the effects of food shortages. It is possible that these groups are able to survive on dissolved organic matter in seawater. Some planktotrophic larvae can increase the chances of survival by cloning or other responses to appropriate environmental conditions. We must continue to emphasize the high mortality rates suffered by planktotrophic larvae from loss by currents, predation, and food shortage. In some species of echinoderms, including asteroid sea stars and brittle stars, larvae have the remarkable ability to reproduce asexually while in the water column. The question is, under what circumstances would this type of response be advantageous? A laboratory study of the common Pacific sea star Pisaster acraceus demonstrated that larvae tended to reproduce asexually by budding when food was more abundant. It would make sense that larvae in the field would bud when food was more concentrated, which would increase the larvae population and, therefore, provide more individuals that might allow survival into the next generation. In a sand dollar species, larvae were found to reproduce clonally in response to chemical cues from fish predators. The clonally produced offspring larvae were smaller, which might have made them less visible to predators. There are now scattered examples of invertebrates that can produce either lithotrophic or planktotrophic larvae. One marine nudibranch revealed an interesting strategy. Starved adults laid eggs that eventually developed into planktonic larvae, but when adults were fed, larger eggs tended to produce some individuals that developed directly, hatched, and had no planktonic stage at all. This suggests that plasticity in development might be a response to food conditions that allows a female to disperse her young away from locally poor food conditions. Much more research is needed to see how this affects the fate of marine populations. The macro scale, major separations lead to biogeographic structure. The geographic range of a species with planktonic dispersal is greater than the range for species without planktonic larvae. Planktonic dispersal gives some species the opportunity to invade distant shores merely by being passengers on the ocean's transport system of currents. We rarely see this process in action, but several species have invaded new coasts in the past 100 years, and their spread has been documented. We mentioned earlier in this connection the shore periwinkle Littorina littoria, which invaded Nova Scotia waters in the late 1800s and has now spread as far south as New Jersey. It stands to reason that current systems that bring water toward shore should also contribute toward increased larval recruitment. We have noted, for example, that the Galapagos Islands stand in a current system that is not conducive to successful larval settlement on the islands. Most coasts are likely to vary much more, owing to variations in current transport. On the west coast of North America, for example, areas of upwelling are often localized, often on a scale of tens of kilometers. When an upwelling system is close to shore, larvae may be transported mostly out to sea in surface waters, which would strongly reduce recruitment. In El Niño years on these coasts, one might expect a great increase in recruitment, since the influx of warm surface water will trap larvae at the California coast and even transport coastal water northward. During El Niño years, the usual isolation, that is, enforced by current systems at Point Conception, California, is broken down and coastal water is transported northward, bringing larvae to the coast of Northern California, often during subtropical storms. Connolly and Roughgarden found that barnacle recruitment increased strongly over a broad region in central and northern California from 1996 to 1997, a probable result of El Niño. Thus, infrequent changes in climate provide opportunities for breakthroughs in dispersal between previously isolated biogeographic zones. While there are some exceptions, invertebrate species with planktonic larvae appear to have far greater geographic ranges than those species with direct release. Figure 7. 31 shows a series of biogeographic zones on the Atlantic coast of North and South America, identified for the distinctness of their respective benthic species. Note that species with non-planktonic dispersal tend to occupy 2 to 3 zones, whereas those with planktonic larvae occupy 4 to 5 zones. Although planktonic dispersal increases the geographic range, planktotrophic larvae with dispersal times of many weeks or months do not seem to have any broader range than those whose larvae spend only 2-6 weeks in the plankton. Molecular genetic variation and other markers can be used to identify barriers to coastal zone dispersal. In Chapter 20, we discuss the large-scale biogeographic structure in the ocean, where groups of species are found in similar widespread biogeographic areas, known as provinces. Such differences would not occur unless there were barriers to dispersal, which many different species have in common. Why would there be such barriers? 2 important factors are involved. First, some physical barriers to dispersal often prevent larvae from spreading. Teleplanic larvae may occasionally traverse broad oceanic distances, as mentioned previously, but most marine planktonic larvae cannot make it across broad oceans such as the Atlantic or the Pacific. Such barriers also exist along coastlines. Separate current circulation systems appear to isolate groups of species north and south of Point Conception in Southern California. South Florida isolates populations of the southeast United States from those within the Gulf of Mexico. On either side of the barrier, one observes closely related species or strong genetic differences between populations of the same species, as can be measured using DNA sequence differences. We might expect that species with long-distance planktotrophic larvae would have coastal populations with high connectivity. Many current systems tend to move parallel to coasts, and these should carry planktotrophic larvae long distances. It is very difficult to study this process directly because larvae are difficult to follow at sea, although this has been done in some cases. Instead, we would seek indirect evidence, such as genetic similarity among coastal populations, as evidence of interchange. This evidence would be especially strong if it is obtained from different genetic sources, such as different genes coming from different parts of the genome. Along the Pacific coast, the barnacle Balanus glandula has a broad geographic distribution, which suggests a great deal of transport of larvae along the coast. It is no surprise, therefore, that there is a great deal of genetic homogeneity of this species in DNA sequences from British Columbia in Canada to Northern California. However, a major difference in DNA frequencies was found between Northern and Southern California. The strong climb coincided with an apparent isolation of current systems that could be demonstrated by simply placing labeled oceanographic drifting floats in Oregon and Southern California. The drifters moved hundreds of kilometers in 40 days but never overlapped each other. The major genetic difference found, therefore, is a reflection of a broad-scale geographic isolation that is probably reinforced by currents. A more recent study on the red abalone, Haliotis rufescens, also shows genetic homogeneity from Monterey, California, to Oregon over an enormous number of single nucleotide polymorphisms. But still, there were a number of genes whose single nucleotide polymorphisms showed significant geographic variation. The online bonus chapter, Molecular Tools for Marine Biology, discusses procedures, but large number of sequences collected could be identified as distinct genes, and those that were significantly variable geographically were involved in hypoxia, energy metabolism, calcification, and other physiological processes in which climate change might come into play as a major selective force now and in the future. Recent geological history may be very important in explaining isolation among populations within species along coasts. There is a rather complex geographical pattern of genetic differences in Southern California in populations of the small harpacticoid copepod Tigriopus californicus. This species lives in tide pools and does not have planktonic dispersal, and we would therefore expect little dispersal between sites by currents. But a very different pattern was found toward the northern part of the Pacific coast of North America. There was very little differentiation at the same genetic locus along the coast as you go northward from Puget Sound in Washington state into Canada and Alaska. This homogeneity derives from the glacial history of the past few tens of thousands of years. It is likely that Tigriopus populations have only recently spread northward after the retreat of the glaciers, and this makes the populations resemble each other genetically along great distances. For species with calcareous shells or skeletons, the trace elemental composition can sometimes be used to trace the origins and connectivities of populations along coasts. For example, planktotrophic larvae of bivalves often have larval shells with elemental compositions that are, in effect, a signature of their point of origin. Thus, larvae originating in different locations along a coastline may have specific compositions that reflect the origin of the larvae. When juveniles are collected, one can examine the elemental composition of the youngest part of the shell and see if it matches the local area or the signature formed by an origin in another area. Using this technique, the California sea mussel Mytilus californianus was found to originate in the northern parts of the coast of Southern California but spread evenly toward the south. This may relate to a spawning, larval period that occurs when currents are moving strongly along the coast. Another mussel species had much more localized elemental compositions, which reflected larval spread during a time of year when longshore currents were not strong, so recruiting individuals reflected more their local point of origin. A second barrier to dispersal is strong natural selection. If a larva disperses to a location where it cannot survive or where its adult phase cannot survive, then individuals at that point are liable to be genetically different and isolated from the
source of those dispersing larvae. A good example is Cape Hatteras in North Carolina. Summer water temperature increases greatly to the south of this cape. It is not unusual for planktonic larvae of the blue mussel Mytilus edulis to be carried by coastal currents in spring to the south of this cape, followed by settlement and metamorphosis into juvenile mussels. But by midsummer, the temperature is simply too high for this species and individuals die. This explains the southerly extent of Mytilus edulis. Recent experiments with planktonic larvae of the eastern United States fiddler Crabuca pugulator show that their larvae cannot tolerate the low temperatures found north of Cape Cod. This may explain the northern limit of the species. A particular type of intraspecific genetic change with distance along the coast can be an excellent indicator of isolation that is caused by natural selection. Consider a hypothetical coast that has a very strong spatial gradient in temperature. It might be that one allele at a genetic locus might be favored at one end and an alternative allele might be favored at the other end of the gradient. Thus, natural selection might be isolating populations on either side of this genetic transition, where the frequencies of variance change over space. As discussed in Chapter 4, this spatial change of variation is known as the cline. Natural selection favors different variants on either end of the cline, which may isolate the two populations from each other if the selection gradient is very strong. If natural selection were operating on two genetic loci, then the strength of isolation would be even greater on the macro scale. Isolation between regions may result in a large group of separated populations of a species which might have only occasional interchange. Because of numerous barriers to dispersal along coasts and across open seas, we would expect that widespread marine species would comprise a series of isolated populations. These populations might be affected by very different local conditions, and population change might be independent in each local region. In the long run, if isolation were never broken, the populations might diverge genetically and separate species might be produced. But occasional exchange of far-traveling individuals might keep the chain of connection open and new species might not be formed. Robert Cowan and colleagues have analyzed the structure of the entire Caribbean basin and have found that currents tend to isolate distinct island groups from each other, creating a series of subpopulations of coral reef fishes. Typically, larval dispersal is on the order of 10–100 kilometers, although many of the subregions are not isolated and receive immigrants in substantial numbers. Still, the adaptations coral reef fish larvae have that cause them to be retained in their natal reefs tend to maintain isolation among the regions into a series of island groups among which there is no major population replenishment from other areas. There also are 4 major regions that are largely isolated: the Eastern Caribbean, the Western Caribbean,